So hello students, we are doing chapter number 6, that is molecular basis of inheritance. Now I'm just revising the previous topic, that is packaging of DNA once again in this class. Now Roger Kornberg 1974 reported that chromosome is made up of DNA and protein. Later, Beadle and Tatum reported that chromatin fibers look like beads on the string, where beads are repeating units of proteins. Now, the proteins associated with DNA are of two types: basic proteins, histones and protamine, and acidic non-histone chromosomal proteins. The negatively charged DNA molecule wrapped around the positively charged histone proteins to form a structure called nucleosome. The nucleosome core is made up of four types of histone proteins: H2A, H2B, H3 and H4, occurring in pairs. 200 base pairs of DNA helix wraps around the nucleosome by 1 3/4 turn and plugged by H1 histone protein. Repeating units of nucleosomes form the chromatin in nucleus, which is a thread-like structure. The chromatin is packed to form a solenoid structure of 30 nanometer diameter. Further supercoiling forms a looped structure called the chromatin fiber. Now the chromatins are of two types. So nucleosome constitute the repeating units, beads on string, which constitute coiled thread like stained structures called chromatin in nucleus. So we have two types of chromatin. One is euchromatin and second is heterochromatin. Now we will see here the difference between euchromatin and heterochromatin. So euchromatin is lightly stained, and the loosely coiled region of the chromatin is called as the euchromatin. It is transcriptionally active region, and in heterochromatin it is darkly stained, the tightly coiled region of chromatin, and it is transcriptionally inactive. So in the diagram also you are seeing here the orange colored regions, which indicates the heterochromatin, and here is the purple color you are seeing here, this is the euchromatin. So this is also, you can see the how it looks like, heterochromatin and euchromatin. So heterochromatin contains inactive genes and euchromatin contains active genes. Euchromatin is transcriptionally active and heterochromatin is transcriptionally inactive. So regions of the chromatin which are loosely packed during interphase are called euchromatin, and regions of chromatin which are densely packed during cell division are called heterochromatin. Now, next is the central dogma of molecular biology. Francis Crick proposed the central dogma of molecular biology, which states that genetic information flows from DNA to messenger RNA, that is transcription, and then from messenger RNA to protein, that is translation, always unidirectionally, except bidirectionally in some viruses, and the process is called reverse transcription. So here is the flow of information from DNA to RNA and from RNA to proteins. This is the central dogma of molecular biology. So it is shown over here. From DNA, by the process of transcription, RNA is formed, and from DNA to DNA, if DNA is formed from DNA, that is replication, and if from RNA there is a synthesis of protein, it is called as translation. Now next is the search for genetic material. So the transformation and the transforming principle. So transforming principle, Frederick Griffith in 1928 conducted experiment on bacteria, that is Streptococcus pneumoniae, bacterium responsible for pneumonia. There are two types of strains of this bacteria. Some produce smooth, shiny colonies, that is S strain, and others produce rough colonies, that is R strain. Mice infected with the S strain, that is virulent, die from pneumonia infection. But mice infected with the R strain, that is non-virulent strain, do not develop pneumonia. So here is the difference between S strain and R strain. So in S strain, it produces a smooth colony on a culture plate. In R strain, it produces a rough colony on a culture plate. S strain produces a polysaccharide coat. R strain polysaccharide coat is absent. S strain is virulent. R strain is non-virulent. So here is the transformation and the transforming principle. So when S strain is injected into mice, mice die. When R strain injected into mice, mice survived. So S strain, that is if heat killed, when injected into mice, then mice survived. And S strain heat killed plus R strain, which was live and injected into the mice die. So Griffith concluded that R strain bacteria have somehow transformed by heat killed S strain bacteria. So some transforming principle transferred from S strain to R strain and enabled the R strain to synthesize a smooth polysaccharide coat and become virulent. So this must be due to the transfer of the genetic material. Now here is the figure which is showing Griffith's experiment on transformation. So how if virulent strain of bacteria, if injected into the mice, it died, and if non-virulent strain, that is the rough strain, is injected into the mice, it survives. Then if heat killed virulent strain is injected into the mice survived, and if heat killed virulent strain plus non-virulent R strain is injected into the mice died. Now next is biochemical characterization of transforming principle. So later Oswald Avery, Colin MacLeod and Maclean McCarthy discovered that DNA from the heat-killed S strain bacteria caused the transformation of R strain bacteria. They also discovered that proteases and RNAs did not affect transformation, while DNAs inhibited the process. So DNA caused transformation of R cells to S cells, that is DNA was the transforming principle. Now next is DNA is the genetic material. The proof for DNA as the genetic material, it came from the experiments of Alfred Hershey and Martha Chase in 1952, who worked with bacteriophages. The bacteriophage on infection injects only the DNA into the bacterial cells and not the protein coat. The bacterial cells treats the viral DNA as its own and produces more virus particles. Now, in one preparation, the protein part was made radioactive with S35 and in the other nucleic acid DNA was made radioactive with P32. These two phage preparations were allowed to infect the culture of E. coli. Now, soon after infection, before lysis of cells, the E. coli cells were gently agitated in a blender to loosen the adhering phage particles and the culture was centrifuged. The heavier infected bacterial cells pelted to the bottom and the light viral particles were present in the supernatant. It was found that when phage containing radioactive DNA was used to infect E. coli, the pellet contained radioactivity. So if phage containing radioactive protein coat was used to infect E. coli, the supernatant contained most of the radioactivity. So this suggests that during infection by the virus, the viral DNA enters the bacterial cells and that has the information for the production of more viral particles. So it proves that DNA and not protein is the genetic material in bacteriophage. So this is the figure which is showing the Hershey and Chase experiment, so in which you are seeing the different steps. So first is infection, second is blending and third is centrifugation. So in this diagram you are seeing how this radioactive S35 is incorporated into the protein capsule, and how this radioactive P32 is incorporated into the DNA, and how the steps, that is infection, blending and centrifugation, is taking place. Now we have the characteristics of genetic material. So first is genetic material is able to generate its replica. Second is it is chemically and structurally stable. Third is it provides scope for mutation necessary for evolution. Fourth is it is able to store genetic information which is inherited. Next we have it is able to express itself in the form of Mendelian character. Now here is the difference between the DNA and RNA, or you can say that is the stability of DNA and stability of RNA. So here in the stability of DNA, the complementarity of the two DNA strands provides stability to the molecule. Now in the stability of RNA, we have this hydroxyl group in RNA is present at every nucleotide, and it makes the RNA labile or unstable, and RNA gets easily degradable. So stability of RNA, we have thymine instead of uracil in DNA provides additional stability, and in stability of RNA we have RNA also act as a catalyst called ribozyme, so hence reactive. Now stability of DNA. Now DNA is more stable chemically and structurally, and it is the preferred nucleic acid for storage of genetic material. RNA mutates faster as compared to DNA as it is unstable. Now we have RNA world, that is ribonucleic acid. So ribonucleic acid was the first genetic material, and essential life processes evolved around it. And the 2'-hydroxyl group of ribonucleotide is a reactive group that makes RNA a catalyst. So it is evident that essential life processes such as metabolism, translation, splicing, etc. have evolved around RNA even before DNA has evolved as a genetic material. So the nitrogen bases are of two types: purines, that is adenine and guanine; pyrimidines, we have cytosine and uracil. So we have ribose sugar plus nitrogenous base, it forms a ribonucleoside. We have ribonucleoside plus phosphate group, it forms a ribonucleotide. Now we have this different types of RNA. We have messenger RNA, we have transfer RNA, we have ribosomal RNA. Now in detail we have the types of RNA. First is mRNA, that is the messenger RNA. It brings the genetic information of DNA transcribed on it for protein synthesis. It is single-stranded. Then we have transfer RNA or soluble RNA. It acts as an adapter molecule that reads the code on one hand and binds to the specific amino acid on the other hand. Transfer RNA has a clover leaf-like secondary structure, but actually it is an inverted L-shaped compact molecule. It has an amino acid acceptor end, that is 3'N, and an anticodon loop where the three bases are complementary to the bases of the codon of particular amino acid. Third type is we have ribosomal RNA. Now before doing the ribosomal RNA, here is the structure of transfer RNA. Now this structure is very important. It is also given in your NCERT book also, and sometimes it comes in the exams also. So here you are seeing the sites, different sites over here. One is amino acid attachment site, yes, yeah, where there is written CCA, 3'-hydroxyl group. Then on the one side you are seeing this T-C loop, and you are also seeing the extra arm that is variable. Then you are seeing here the anticodon loop, and then you are seeing here the DHU loop, and on the 5' end you are seeing the phosphorylated 5' terminus. So this is the structure of transfer RNA. Now third type is ribosomal RNA. So it forms the structure of ribosomes. It also plays a catalytic role during translation. Now next we have DNA replication. So Watson and Crick suggested that two strands of DNA separate from each other and act as template for synthesis of new complementary strands. So after the completion of replication, each DNA molecule would have one parental and one newly synthesized strand. So this method is called semi-conservative replication. So now here is the proof for semi-conservative replication of DNA. Matthew Michelson and Franklin Stahel have performed an experiment using Escherichia coli to prove that DNA replication is semi-conservative. They grew E. coli in a medium containing NH4Cl until N15 was incorporated in the two strands of newly synthesized DNA. This heavy DNA can be separated from the normal N14 DNA by centrifugation in cesium chloride density gradient. They then transferred the cells into a medium with normal NH4Cl ammonium chloride and took out samples at various time intervals and extracted DNAs and centrifuged them to measure their densities. The DNA extracted from the cells after one generation of transfer from the N15 medium to N14 medium, that is after 20 minutes, had an intermediate hybrid densities. So the DNA extracted from the cells after one generation of transfer from the N15 medium to N14 medium, that is after 20 minutes, had an intermediate hybrid density. And the DNA extracted after two generations, that is after 40 minutes, consisted of equal amount of light DNA and hybrid DNA. And similar experiment was conducted by Taylor et al. In 1958 by using radioactive thymidine. They proved that DNA on the chromosomes replicate in a semi-conservative manner. So here is the diagram which is showing the Michelson and Stahel's experiment. So here you are seeing the DNA extracted from the cells after one generation of transfer from the N15 medium to N14 medium, that is after 20 minutes, had an intermediate hybrid density. And the DNA extracted after two generations, that is after 40 minutes, consisted of equal amounts of light DNA and hybrid DNA. Next we have the process of replication of DNA, and first of all we are here studying the enzymes for DNA replication. The main enzyme involved is the DNA dependent DNA polymerase. It catalyzes the polymerization of deoxynucleotides on DNA template. Next we have helicase. It unwinds the DNA strand to form the replication fork. Then we have DNA ligase. It joins the Okazaki fragments which are formed on the lagging strand. Then deoxyribonucleoside triphosphate serve dual purpose. It first of all acts as the substrate and it provides energy, the polymerization reaction. So sometimes it comes in the 2 marks questions that deoxyribonucleotide serves dual purpose, and what are these? So you will write it acts as the substrate, and other is it provides energy for the polymerization reaction. Now next we have process of replication of DNA. Replication of DNA occurs in S phase of the cell cycle DNA replication is a process of producing two identical copies of DNA from a single DNA molecule and is a process of biological inheritance. DNA replication begins at a unique and fixed point called origin of replication, that is ORI. DNA replication is an energy expensive process. So here you are seeing the replication fork. Yes, Y-shaped structure. So in this you are seeing here the two strands. Now they are uncoiled, unzipping of the strands are there, and you are seeing here the enzyme topoisomerase. Then you have here the helicase. Then you have single strand binding protein SSBP. So then you are seeing here the two strands, they are separated out, and on these two separate strands there is the formation of new strands. One is the leading strand and other is the lagging strand. So you are seeing here leading strand is continuous and lagging strand is discontinuous, and it is also called as the Okazaki fragments. Small fragments are there, and these Okazaki fragments are further joined by the enzyme, that is the DNA ligase. Now here this is the diagram which is given in your NCERT textbook. So here is you're seeing the Y-shaped structure. This is the replication fork. So in this the two strands are there, template strands, and here you are seeing the newly synthesized strands, that is the leading strand, and the other is the lagging strand. And the lagging strand you are seeing here, this is in small fragments which are called as Okazaki fragments, and these Okazaki fragments are further joined by DNA ligase to form a complete strand. Now what are the steps of DNA replication? So first is initiation. The complementary strands of DNA double helix are separated by two enzymes, DNA gyrase and DNA helicases. This is called unwinding of double-stranded DNA. The unwinded strands of DNA are stabilized by proteins called single-strand binding proteins. Unwinding of double-stranded DNA forms a Y-shaped structure which is called replication fork. Next step is elongation. An enzyme called primase initiates replication of the strand oriented in the 3' towards origin or 5 towards 5' direction, and this generates 10 to 16 nucleotide long primer RNA. The DNA-dependent DNA polymerases catalyze polymerization, that is 2000 base pairs per second, of the nucleotides only in 5' to 3' direction. The replication of this 3' to 5' direction is continuous and is called the leading strand, while the replication of the second strand, that is from the 5' to the 3' strand, it is discontinuous and is known as lagging strand. So Okazaki fragments are formed, and these fragments are joined together by the enzyme called DNA ligase. Now the difference between leading strand and lagging strand. So here are some differences which you are observing over here. So leading strand, it is formed continuously as a single fragment, and in the lagging strand, in the beginning, it is formed in the form of small fragments called Okazaki fragments. So it requires, in leading strand, it requires only one primer to initiate the growth. Even in the lagging strand, you see here every fragment requires separate RNA primer to initiate. Now in leading strand, DNA ligase not required. Now but in the lagging strand, DNA ligase required to join the different DNA fragments. So direction of growth is from 5' to 3' direction in the leading strand, and direction of growth of complete strand, that is 3' to 5', and however for the Okazaki fragment it is 5' to 3' direction.